Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Automated Mass Spectrometry Workflows for High-Throughput Bioanalysis of Proteins with SISCAPA. SISCAPA is a patented sample presentation workflow that purifies analyte peptides directly from crude sample digests. Used along with mass spectrometry, this technique allows quantification of large numbers of proteins in tissues and body fluids with precision, sensitivity, and high throughput. Our webinar panelists are here to describe a highly automated SISCAPA-MS system and discuss a number of applications of this technology. So let's meet our panelists. Dr. Lee Anderson is the founder of SISCAPA Assay Technologies. Lee is going to discuss in detail the topic of today's webinar, that is, automated mass spec workflows for the high-throughput bioanalysis of proteins with SISCAPA. Rachel Bolger serves as a product manager at Agilent Technologies. Rachel will describe automated solutions for protein analysis workflows. I am John Sterling, editor-in-chief of GEN, and I'm going to serve as the moderator. After the panelists make their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible. So if everyone's ready, let's get going. Dr. Lee Anderson will be our lead-off presenter. Lee, the floor is yours.
Thank you, John. I'd like to begin by talking about the essential character of this workflow. This is, as John mentioned, a workflow that is designed to purify analyte peptides and to purify them directly from crude sample digest, enabling an extremely simple approach to the generation of effectively pure samples for mass spectrometry. The addition of internal standard labeled peptides, stable isotope labeled synthetic versions of the analyte peptides we're measuring, allows us to do internal standardization, effectively an isotope dilution method for quantitation that allows us to achieve very high precision in the relative quantitation of the peptides extracted from such sample digests. The workflow is basically an extremely simple approach in which a protein sample, for example, human plasma, is digested with an enzyme like trypsin to yield a large collection of peptides. The stable isotope labeled internal standard version peptides are spiked into that digest, and then antibodies specific to the peptides we've chosen to analyze are bound to an affinity support and used to bind out of the digest with the internal standard spike the peptides of interest. Everything else is washed away, and then we can elute the desired analyte peptides for introduction into the mass spectrometer. The technique is particularly designed to allow us to do protein quantitation, and quantitation without the interference of a number of other features that are present in physiological samples, which are due to, for example, protein-protein interactions. The objective of digesting the sample as the first step in the protocol is effectively to eliminate protein-protein interactions, and this removes antigen-antibody interactions, protein receptor interactions, etc. And therefore, this allows us to do unencumbered measurements of total protein in the sample. But I want to emphasize that, therefore, we are not measuring protein-protein interactions. And if one wanted to measure, for example, the fraction of a protein that was bound to a receptor, that needs to be measured by other techniques, including ligand-binding assays. This approach is focused specifically on the measurement of the quantity of the protein. Now, mass spectrometry is emerging as a very important method of doing quantitative measurements of proteins. And this is primarily because it overcomes the major limitations of the immunoassays, which have been conventionally used to measure protein abundance. And the issues in which mass spectrometry can make a major improvement over immunoassays have to do with specificity, the available use of the internal standard labeled proteins to give real internal standardization, easy multiplexing and rapid configuration of the assays. Now, the specificity, for example, is an extremely important feature of mass spectrometry. because using MRM mass spectrometry or methods which allow us to focus on the specific mass of the expected of the peptide and masses of particular fragments of it, allows us to get effectively absolute molecular specificity for the analyte. And as I'll mention in a moment, this is something which is not directly possible by immunoassay. The inclusion of the internal standards, as I've mentioned, allows us to have an internal standard against which to do the quantitation, which once again is not possible in an immunoassay. And we can do multiplexing because mass spectrometry is uniquely able to measure many things without inherent interferences between them. And the methods allow rapid configuration of the assays. Now, all of this is possible and relatively easy to achieve if the protein can behave as if it were a small molecule, which is the area in which mass spectrometry has historically primarily been used. And this is the reason why we digest the proteins to peptides, effectively to render them small molecules for the purpose of mass spectrometry. Now, this slide shows a series of specific peptides which have been selected to represent the major proteins present, about the top 50 proteins present in human plasma. And in each of these cases, we can find multiple tryptic peptides which are unique to the specific protein that we're attempting to measure here. And we can measure those by MRM, multiple reaction monitoring techniques, using a triple quad mass spectrometer in an unfractionated tryptic digest of human plasma. And this is work that I did some years ago with Christy Hunter at AB-Sciax. And this was aimed at measuring a series of proteins which are of some clinical interest but are present at high abundance. Now, the caveat is that this approach using an unfractionated digest and a mass spectrometer using the liquid chromatography separation in front allows one to access really only proteins that are present at a level above a microgram per mil in plasma. And the technology, in order to get sufficient sensitivity, requires an extended analysis time in terms of the liquid chromatography gradient in front to be able to resolve a large number of peptides and measure them individually. So that limitation is a significant one from the point of view of measuring biomarkers and other biological molecules. Therapeutics, for example, we'd like to be able to measure in complex matrices. So SISCAPA was designed to address that limitation, those limitations very specifically. And in particular, I'm going to indicate why this allows us to improve the sensitivity of the assays to go far below a microgram per milliliter level. We can simplify the workflow very significantly, and we can increase the throughput and therefore the cost of doing the analyses very substantially using the SISCAPA workflow. Now, in the first place, the sensitivity is substantially enhanced because, in contrast to the workflow I just showed in the top panel, which involves the chromatographic separation of all the peptides in a very complex digest, a small amount of a complex digest, to look at all the peaks. We can, in fact, with a specific antibody against one peptide, in this case shown as attached to magnetic beads, go into a much larger volume of sample digest than could ever be practically loaded onto the chromatography system going into the mass spectrometer directly. And we can pull effectively all of the analyte peptide out of that much larger volume of sample digest and present that to the mass spectrometer. That leads to a very significant increase in the sensitivity of the assay because, in fact, we're simply generating more of the analyte from a larger sample. And since what is practically injectable into an LC system at this point for the kind of mass spectrometry we use for quantitation is on the order of 0.1 microliter of plasma's worth of a plasma digest. If we can, in fact, capture analyte out of a milliliter of digested plasma, which in fact has been done and is practical, we can generate a much larger volume of sample to introduce into the mass spectrometer, increasing the sensitivity. At the same time, by removing the other peptides, the matrix peptides, of which there are literally hundreds of thousands of different peptides in a complex sample digest such as plasma, by removing those, we end up with a much, much simpler sample. And this is a sample which is getting close to the ideal analytical sample, the analytical situation of analyzing effectively nothing, effectively a pure sample of the peptide we want, which is very low abundance in a typical case for most biomarkers and other molecules we're looking at. And that means we can get by with much less chromatography and a much shorter cycle, so that increases the throughput very substantially and also improves the general performance of the LC system significantly by virtue of the fact that they're not overloaded. Now, I want to emphasize in one slide the real objections to immunoassays that this kind of an approach allows us to achieve. In an immunoassay, there are a variety of mechanisms by which interferences can generate false negative or false positive results. In fact, we can have situations in which there are complexes with proteins, unsuspected proteins that block an epitope and prevent generation of signal. We can have autoantibodies to a particular protein that block epitopes and block generation of a signal. Or in fact, the protein target could be cleaved, eliminating one of the epitopes. And conversely, we can have false positives generated by autoantibodies as well. Now, the key thing about these mechanisms of producing artifactual results is that there is not in an immunoassay any little molecular microscope in the system, which allows you to actually visualize what it is that is generating the signal, whether it's really the analyte you're expecting or not. And those assay interferences, therefore, are extremely difficult to debug and often difficult to recognize. And the use of mass spectrometry allows us to transcend those difficulties because we are in a position to be absolutely certain of what it is we're measuring and what the molecular structure of that analyte is. Now, the assays of using this kind of workflow, I should mention, in fact, are already in clinical use. In particular cases where such interferences, as I've just described in an immunoassay, turn out to be prevent generation of the correct clinical result, which is a very high priority. And this is an example for an assay for the protein thyroglobulin, which is used in the detection of the recurrence of thyroid cancer. This is a system which was looked into in some detail by Dr. Andrew Hoofnagel at the University of Washington, who realized that autoantibody interferences in this immunoassay system were a significant problem because the autoantibodies occur in about 20 to 40% of the patients that one wants to measure for recurrence of thyroid cancer. And he realized that using the SISCAPA approach would eliminate these interferences because the tryptic digestion step will digest all the autoantibodies to peptides, and they won't therefore interfere with the detection of a specific thyroglobulin assay peptide. Now, this assay has been adopted at the Mayo Clinic recently and at ARUP in Salt Lake City, and it's been used to run tens of thousands of clinical samples and has been shown to give the correct result in situations where the autoantibodies prevent the conventional immunoassays from working. So this is an approach which actually has turned out to be successful and clinically relevant in eliminating the interferences which plague the immunoassays. So the selectivity of SISCAPA assays are enhanced by the fact that the antibody capture reagent can pull out very specific peptides. And in fact, for measurement of molecules like therapeutic antibodies, it's possible to pull out specific peptides from the variable regions of those specific antibodies, which are going to be unique to particular clones. And we can pull out, we can measure recombinant proteins, obviously, versus patient-derived proteins as immunoglobulins by focusing on the clonal specific peptides, the CDR region peptides from those antibodies. And at the same time, we can usually almost always distinguish human versions of proteins from those that occur in other species, obviously relatively straightforwardly in rodents, and in most cases also in primate species as well. In looking at how one would use this approach to measure peptides that are specific to particular antibody sequences, we can leverage the fact that there are large regions of sequence in an antibody that are frameworks or constant sequences common to large families of antibodies, and then there are the complementarity determining regions which have specific sequences which are particular to the clones we may want to measure as a therapeutic, or in fact potentially as a tumor. And a variety of those are shown in the right-hand panel of this slide, which lists a series of tryptic peptides which are specific for the framework regions of a variety of specific isotypes and chains of human immunoglobulins. And on the left-hand side is an example from one of the therapeutic monoclonal antibody products of a peptide which is specific to that particular clonal product and can be used to measure the amount of that specific therapeutic antibody administered to humans. So we have a variety of methods of measuring classes of antibodies and of specific clones of antibodies which can be applied in this approach. The value of doing this approach for the pharmacokinetics of biologics has to do with the fact that in many cases ligand-binding assays or immunoassay type approaches can be confounded by the presence of soluble target, which can bind to the antibody molecule, and anti-drug antibodies. Similar to the situation with the thyroglobulin assay, these can interfere with detection, and they may do so in a patient-specific manner, which of course is more difficult to detect during the development of an assay and to prevent that from happening when large numbers of patient samples are looked at later. And this is even more complicated for cases in which antibody-drug conjugates are used, because one may have the confounding of the antiprotein epitopes that may be used in the ligand-binding assay by the drug payloads themselves, and the heterogeneity of that payload can also confound the measurement. So SISCAPA is capable of eliminating these problems because of this fact that we digest the protein and we can skillfully select which signature peptides we're going to use in the quantitation. For high-value biologic drug candidates, developing reagents to do the detection of specific variable regions of immunoglobulins is a matter of extreme emphasis at this point, and is something which is generally possible. While it's not possible to guarantee that there will always be a unique tryptic peptide in the CDR regions of a particular immunoglobulin, it appears to be relatively frequently accessible, and if necessary, other digestion enzymes can be used. It's obviously very selective because of the sequence specificity of the peptide detection for the drug of choice, and these can be used across platforms, as I mentioned, including animal studies, clinical trials, and for therapeutic drug monitoring. I wanted to show an example of the use of the genomics databases to look at the capability of predicting whether a particular peptide is likely to show up as a confounding factor in a particular species. And by simply doing blast searching against the databases, the sequence databases, which are available and currently growing for a number of species, we can predict whether or not a given tryptic peptide is possible to be produced in proteins of a particular species with relatively great certainty. So this tells us the spectrum of species in which an assay based on a particular peptide is going to work. And conversely, it allows us to define species in which that peptide should never occur in any protein. And this gives us a wide variety of good opportunities to make analyte-free matrices, which is extremely useful in developing calibrators and doing dilution experiments to characterize the assays themselves. So this is something that can be predicted successfully directly from the genomics databases. So I'd like to talk next a little bit about the technology and the development of the assays themselves, specifically and focus on the reagents and the platforms used for doing the assays and the simplicity of those, the custom assay development options, and the approach to technology access using this patented method. As I've described the workflow, each SISCAPA assay contains only two assay-specific components. One of them is a high-affinity antibody, which has been created specifically to bind the tryptic peptide that we've selected as the surrogate we're going to use to measure our target protein. And the second component is a stable isotope labeled version of that same peptide, same sequence, exactly the same chemical structure, but labeled with stable isotopes in such a way that the mass spectrometer can distinguish it from the analyte. And these two components, the peptide is relatively straightforward to create by synthesis, but the antibody, particularly as we generally use them, monoclonal antibodies to the specific peptide targets, are something which we've had so far a significant amount of experience in generating. On the order of hundreds of these antipeptide antibodies have been made, and the process for doing it is relatively well understood at this point. SISCAPA Assay Technologies offers this service as a contract service using a reagent pipeline that we've developed over the last 6 or 7 years. And that process begins with the selection of the specific peptides that are going to be used to measure proteins, to optimize their antigenicity, obviously the ability to make the antibody, the mass spec performance of the peptide, and the stability of the reagents and the system that we use for doing the affinity enrichment. The antibody development process includes the selection and purification, either of polyclonals or monoclonal antibodies, as we typically use, generation of the labeled peptides, and optimization of the mass spec MRM, typically parameters used for the detection of those. And the combination of these allows us to characterize and lock down an assay in a very straightforward manner. As I mentioned, the workflow is meant to be simple and robust at this point, and that is achieved through automating effectively all steps of the protocol using a liquid handling robot, the Agilent Bravo robot platform, as is shown in the lower left here. There are a variety of implementations which have been explored, and the Bravo approach is by far the most robust and simplest at this point. And it has been condensed to a workflow using the 96-well plate format and optimized as an addition-only digestion protocol combined with the affinity isolation of the specific peptides using a magnetic bead processing approach. And so we're going to attempt to avoid anything which resembles a step in which part of the analyte could be lost. And the sample addition only protocol for doing the digestion is key to that approach. The approach that we're using adds the sample to denaturates, carries out reduction and alkylation, tryptic digestion, and quenching of the trypsin itself by addition of liquid reagents directly to the same plate of sample. And then internal standards to label peptides are added. The SISCAPA antipeptide antibodies on magnetic beads are added. Those are incubated. And then the bound peptides on the antibodies on the beads are washed and eluted and placed into the mass spectrometer. The simplicity and the cleanliness of those samples allows us to do most of the experiments, typically running 5 to 10-plex assays with a very short 3-minute chromatography protocol. And as I mentioned, a key feature of this is that there is no solid phase extraction, no precipitation, no filtration, no buffer exchange, centrifugation, or other off-robot sample steps. The only things which are carried out, in fact, off the deck of the robot are the longer incubations when a longer digestion time is required, for example. So using robotic liquid handling, we can do very high throughput processing of samples using a piece of software, which is an implementation of the Bravo's standard workflow control software, which has been optimized for the SISCAPA workflow. And this is an indication of the first user screen, which shows that the 3 component steps of the process which are used to process the samples. The first is the digestion, the second is the capture and enrichment, and the third is the washing and elution of the peptides from the beads. And these three processes are carried out sequentially, and each is a completely self-contained robotic process. This slide shows the user screen from the first of those and indicates that, in fact, the user need only specify two things in this situation: the volume, in this case of plasma, which is going to be processed in each of the wells, and how many wells of plasma is going to be processed. The schematic there shows what labware is placed on each of the pads on the deck of the robot, and after pressing a start button, the process is completely automatic from that point forward. Using this approach, we've been able to very carefully characterize the precision of this workflow, including the digestion, the tryptic digestion step, which had been a significant concern for many people in workflows of this kind. We have looked at this in a stepwise procedure, looking at the coefficient of variation for three assays in this case, across replicate analyses of three different peptides representing three different proteins at different levels of abundance in plasma. And suffice it to say that the CVs, in fact, are extremely low, well competitive with immunoassay performance. And in fact, the difference between the coefficient of variation of the entire workflow, in the bottom three lines there, is very comparable to the coefficient of variation of the mass spectrometric measurement replicate injection measurements of the same sample, as shown on the top three lines, indicating that the effect of the imprecision contributed by the tryptic digestion is very small, typically on the order of 1 or 2% CV of the digestion step itself. I also should mention that since the assays, in fact, do not involve interactions between antibodies, there isn't really a physical mechanism for one SISCAPA assay to interfere with another assay. And therefore, one should be able, and in fact it turns out one is able, to run multiplexed assays, and mix and match multiplexed assays without any interference between them. And as shown in the paper by Whitaker, one gets the same performance of a SISCAPA assay, whether it's run as an individual assay or, in this case, in combinations up to 50 plexes. A key advance in moving this technology towards a robust clinical implementation or a high-throughput experimental implementation is the realization that with this kind of an approach, we can get sensitivities with a robust higher flow implementation, in this case 400 microliters a minute into the mass spec source, using an Agilent 6490 with an ion funnel, and get sensitivity which is comparable to the earlier work with nanoflow chromatography systems. And therefore, this puts us in the regime of doing high-throughput analyses in robust chromatography systems, as are used for very high-throughput work in the small molecule world. In fact, that can be pushed to even higher throughput using a direct injection approach like the Agilent RapidFire, which in fact uses no chromatography and traps the peptide sample on a C18 trap, after which all the peptides are eluted in a single step, a 2-second wide peak into the mass spectrometer. And this works with SISCAPA because of the fact that we are isolating specific tryptic peptides, and therefore there's almost no matrix associated with these samples. It's almost pure analyte. And this significantly reduces the ionic interference that would occur by injecting large numbers of analytes in such a concentrated peak, and allows us to do accurate quantitation at very high throughput, in this case with rates up to 7 seconds a sample. One final thing I'd like to mention is the potential for use of unconventional samples. Because we are denaturing and digesting all the proteins as the first step in the protocol, we can use samples that have, for example, been dried. And dried blood spot samples, which are prepared by effectively taking drops of blood on filter paper and drying them, has proven to be extremely significant from the point of view of a very robust and easy, inexpensive collection of samples, extremely easy long-term storage, and represents an ideal situation for doing large epidemiological and pharmaceutical studies over significant periods of time. So we've looked extensively at the application of these assays in dried blood spots, and this slide shows a couple of panels of interesting clinical analytes which we have developed using the SISCAPA protocol, and which turn out to have very good performance characteristics when applied to dried blood spot samples. This is such a case in which we have looked at a series of historical dried blood spots, 104 individual samples collected over almost 5 years from one individual, and looked at, in this case, two particular proteins. One is the C-reactive protein, and another one is the LPS-binding protein, both of which are acute phase reactants. And in this data, we can see, A, that the baseline levels are extremely stable over long periods of time from samples such as this. But we can also see clear evidence of actual physiological effects. There are 3 yellow boxes there which indicate basically adenovirus infections, nose colds, that this individual experienced, and a red box which shows a period of time during which that individual had a case of pneumonia. And these biomarkers are extremely sensitive to infectious disease events like that. And in fact, in pneumonia, the CRP was increased more than 350 standard deviations from this very tight baseline. Obviously, this is an extremely powerful and successful example of a biomarker, and if all biomarkers could produce differences this large, the field would be an easy one to work in. But this is a clear example of the value of dried blood spot specimens and the power that they represent for detecting long-term trends in biomarkers. Even short-term effects can be extremely interesting to look at. This is a larger series of proteins looked at on a daily basis during the resolution of a bacterial infection, and shows clearly that there are different time courses represented by different biomarker proteins in this case. And as the analysis of this proceeds, it's turning out to represent an opportunity to discover very interesting mechanistic differences that occur over time. Many proteins, in fact, most of them, turn out to be very stable within an individual, and stable in an individual with exhibiting much less variation than there is between individuals. And this is a slide which shows examples of a series of sequential dried blood spots compared for 4 different people. Each person is a line of a different color there. And it indicates that the differences between people are very substantial compared to their differences within a person over time. And that allows one, among other things, to effectively fingerprint individuals and their samples based on the relative levels of their individual proteins constant over time. And this approach allows us to do an extremely precise approach in estimating what the normal variation range of a person is, and that substantially increases the sensitivity for the detection of changes in that level, as contrasted to what is normally done in looking at whether or not a person is deviating outside the normal range of the population. And lastly, I want to mention that the methods that I have discussed, because of the fact that they involve triptych digestion of the sample at the beginning, in fact, are very widely applicable to related types of samples. And this, the graph on the right here shows a comparison of the levels of a whole series of proteins measured in whole blood or in plasma. And for almost all of the analytes, the results are indistinguishable between whole blood and plasma, as they should be, because most of these assays are measuring plasma proteins. Now, there are a series of proteins, a small number of proteins, for which the results are discordant, and it turns out that that's for exactly the reasons that we would expect. Hemoglobin, for example, is very highly concentrated in red blood cells and should not be present in plasma, so it's a point that's far displaced from that line of equivalence. The von Willebrand factor and myeloperoxidase, for example, are strongly associated with other cellular elements of the blood, and therefore give us a different total concentration if we look at whole blood as compared to plasma. But otherwise, the proteins that in fact are segregated, particularly in plasma, which represents the majority of biomarker proteins currently used, give equivalent results in those two samples as we would expect. This is extremely encouraging for the broad applicability in a range of sample types of this kind of technology. So I'd like to conclude by just summarizing the key features of the SISCAPA workflow in terms of its application in measuring protein therapeutics and in biomarkers. Key issue is that it overcomes major weaknesses of immunoassays, and for this reason we're seeing gradual replacement of a number of ligand-binding or immunoassays using this kind of an approach to get around specific interferences generally. The approach is extremely simple and has been automated in a manner that allows us to achieve very high throughput on automated platforms, including particularly the Agilent Bravo. And a number of applications, in fact, are already in place, including the use in laboratory-developed tests for real clinical measurement, in which cases the approach is proving to be very robust and significantly better than the immunoassays that it's displacing. SISCAPA assay technologies provides access to this technology at multiple levels, and therefore it's something which is capable of being adopted in a variety of environments at this point. So I'll conclude at that point and turn it back to you, John.
Lee, thank you so much for a great presentation. You really showed how SISCAPA-MS can provide a powerful approach for targeted protein quantitation. You covered a lot of territory in exquisite detail, so much thanks. If you're just now joining our webinar, welcome. As I mentioned earlier, we'll be conducting a Q&A segment following the panelists' presentations. Please type your question for any or both of our panelists into the Ask a Question box on the lower left of your console, and then hit Submit. Our second panelist, Rachel Bolger, is ready to begin her presentation. So, Rachel, I am turning this over to you.
Thank you, John. My name is Rachel Bolger, as he said. I'm the product manager for Agilent's AssayMap Automated Solutions. AssayMap is one of Agilent's sample preparation platforms. Lee presented today a contemporary approach for addressing challenges faced with protein analysis. The SISCAPA technology is a simple, elegant, robust solution that leverages automation, a new approach to software interfaces, and embraces an integrated workflow concept to create an intelligent, reproducible, high-throughput solution for quantitating by mass spectrometry therapeutic proteins and circulating disease markers. I'd like to take a few minutes to discuss this approach and share with you some of the things Agilent's doing in this space. Protein analysis has undergone a revolution largely due to advancements in mass spectrometry and chromatography. This enables scientists to make quantitative measurements with unprecedented sensitivity and precision. As this field matures, appreciation for the power of mass spectrometry is growing, and demand to fully adopt it is increasing and expanding into new spaces like the clinical laboratory. As a result, demand for greater analytical precision and throughput is increasing concurrently. Despite these innovations, advancement in sample preparation in front of mass spec analysis has failed to match pace. Methods often have tedious pipetting steps and can be imprecise. Attempts to scale are often met with increases in variability due to compounded error. That variability can cloak significant data and result in delays. Challenges such as these can stifle the complete adoption of mass spectrometry. To fully enable mass spec, the challenges of sample prep must be addressed. Lee presented a specific, compelling, and enabling workflow. Agilent's focused on addressing the limitations of sample preparation methods and driving innovation to enable mass spec analysis. I like to think of protein analysis workflows as falling into three fundamental categories: the steps involved in preparing a sample for analysis, the separation steps using HPLC in columns, and the mass spec analysis steps. Innovations are happening across the spectrum here. With R&D efforts and new technologies in all of these categories, Agilent's uniquely poised to consider and develop total end-to-end workflow solutions from sample through to analysis. But I want to share a bird's eye view of what we're doing specifically in the sample preparation space. Lee's SISCAPA workflow is built on the Bravo liquid handling platform. This is a small footprint, state-of-the-art liquid handling instrument that has a proven track record for automating workflows. Expanding on that platform is Agilent's assay map technology, where chromatography meets parallel processing. We've converted the head of the Bravo and equipped it with 96 precision flow control syringes that each connect with a 5 microliter packed resin bed cartridge. We've essentially turned this instrument into something very much like 96 mini HPLC columns to deliver quantitative binding and elution in small volumes. The assay map platform doesn't replace HPLC, but rather brings that level of quality, enrichment potential, and reproducibility upstream into the sample preparation space. The Encore Multispan is a next-generation advanced liquid handling platform with independent X and Y axis pipetting motion that enables tube-to-plate transfers and simultaneous hit picking. RapidFire is a high-throughput solid-phase extraction solution for quantitating on mass spec when liquid chromatography separation is not required. Answers are delivered in seconds. With each of our platforms, we're creating a new way of interacting with automation. Agilent automation is driven by intuitive, interactive, visual interfaces that are easy to use. Our software is designed with attention to complete workflows. Standardized forms create a consistent user experience, and most importantly, our software is designed for analytical chemists, so you can prepare your own samples and perform your own analysis without engaging an automation engineer. This example shows the interface for an assay map peptide cleanup application designed to desalt peptides prior to mass spec analysis. The left side of the form shows the key assay parameters. There are toggle buttons to turn on or off each step. There are inputs for controlling volume and flow rate of liquid through the assay map cartridges, so target binding can be optimized. This provides the flexibility to use this protocol across many different assays without having to reprogram your automation. The center section communicates the deck layout and labware selection, and the right section shows the functional buttons. This interface layout is conserved for all assay map protocols. Software is a powerful component in a workflow solution. It's the means for method transfer and scale. This is a good example of the attention we're giving to software design. But quality solutions don't stop at well-designed workflows. Attention should be paid to the complete spectrum of scale. Already designed into each of Agilent's automation platforms is a growth path that takes you from semi-automated solutions that have a few user touch points to solutions that incorporate robotic arms and integrate all the required components of a specific workflow, such as incubators and centrifuges. So when your throughput needs scale and it's time for complete automation, complete walk-away automation, so too scales the options that are available to you. So true innovation in protein analysis space is not about simply automating methods. It's not about taking plate methods and writing a protocol that automates its use. It's much more than that. It's about concatenating individual applications into an integrated, powerful workflow. It's about reducing the opportunities for human variability. It's about designing creative, intelligent software that's intuitive and easy to use. It's about creating simple, out-of-the-box solutions. And it's about enabling highly reproducible methods that are transferable, robust, and can scale. These are the concepts that are driving Agilent development for protein analysis solutions. These are the values embraced in our designs. And it's through strategic partnerships like ours with SISCAPA team that we're able to drive innovation and provide workflows for mass spectrometry analysis. I want to thank Lee Anderson and the SISCAPA team for their presentation, and I want to thank you for your attention. Now back to you, John.
And thank you, Rachel, for talking about protein analysis workflows and especially showing how they can be best automated. I think your discussion perfectly complemented Lee's presentation, so thank you so much. We're almost ready to begin our Q&A presentation, but first I'd like to ask the audience to disable their pop-up blockers because a short survey on this webinar will be appearing in a moment. We would very much appreciate your feedback on this webinar. So we've had some interesting questions. Let's take a look. And again, we'll take the first question from the audience. So we have our first question that's come in, and it's for Lee Anderson. Can we use this technique for detecting and characterizing amyloid directly in tissue sample, and in parentheses, fresh tissue or paraffin-embedded tissue or tissue in formalin, without microdissection of Congo red positive areas in tissue sections? Lee?
Yes, I believe that would work very effectively, provided that one knows the specific peptide or peptides that you want to detect. The ability of the SISCAPA antibody to enrich the particular peptides out of a digest, whether it's of liquid sample like plasma or whether it's a digest of deparaffinized embedded tissue, for example, is a very straightforward process that seems to work very well.
Okay, we have another question, Lee. What will be needed to evolve from LDTs to CDX, which involves use across labs, technicians, and even platforms?
Well, the SISCAPA approach actually has been validated across platforms in a variety of research labs so far with highly comparable results. And so the primary barrier at this stage in evolving from the LDT use to in vitro diagnostic FDA approvable use is the development of FDA approvable mass spectrometers. These are under development and about to be registered by a variety of the different mass spec platform makers. And once FDA cleared instruments are available, the procedures and reagents that have been developed for LDT use are directly translatable into in vitro diagnostics.
Thank you very much. Another question, Lee. Which MS model is working, I guess, or will work with this approach, or might work with this approach?
Well, we've done it with a variety of different triple quadruple mass spectrometers. The work that I was talking about here was done with an Agilent 6490, which is the high-end triple quad that they produce and is capable of doing very high sensitivity detection at normal flow rates, which is important for the robustness of the procedure in a very high throughput use. But we've used triple quads from a variety of different manufacturers, as I've said, and in fact other kinds of mass spectrometers as well, including multi-TOF instruments.
Great, thank you. And Lee, do you think it is practical to replace the short LC-MS separation in SISCAPA with MALDI-MS?
Yeah, to amplify in this context a little bit from what I just said, the fact that SISCAPA antibodies effectively isolate the target peptide in many cases, and reducing the background to near zero means that's sufficient specificity to allow you to get away with a single-dimensional mass spec analysis as the detection modality instead of the two-dimensional typical MRM approach used in a triple quad. And for that reason, MALDI is an effective way of doing the quantitation for a variety of peptides. I think the key thing in MALDI is the sensitivity that's achievable, and in MALDI, the tryptic peptides that end in arginine are 5 or 10 times more signal generating than lysine peptides. So we've had extensive experience in doing that with arginine peptides, and in many cases those assays appear to perform very well.
Perfect, thank you. Rachel, question for you. Why would you use LC instead of the RapidFire? What would make you use LC when RapidFire is 30 times faster?
Yeah, thank you for that question. And while I'm not an expert on the RapidFire technology, RapidFire is useful when separation is not a requirement in your sample. So that's the area that you'd choose to use it.
Okay, thank you very much. Lee, we're going to go back to you. Give me a second here with these questions. Why would you ever want to capture a peptide instead of the whole protein?
That's a really good question, because obviously many antibodies exist to be able to capture whole proteins. The primary reasons are that if you capture the whole protein, the antigen-antibody interaction that you're using there is subject to all the same interferences that plague the immunoassays that can get in the way of the antibody recognizing the protein and avoid its being blocked by other kinds of interactions. Secondarily, the fact is if you capture the whole protein, you're going to generate on average, for an average size protein, 50 tryptic peptides, whereas we're going to be analyzing 1 in the limiting case. And so the sample is still significantly more complex if you do it that way. And finally, I think while we've been able to demonstrate explicitly on several occasions that we can multiplex the peptide level capture SISCAPA assays up to 50-plex, I don't believe people have explored in any significant way the multiplexing of the whole protein capture approach. And the reason is, there are a variety of reasons. The primary one is the potential for cross-reaction and the other idiosyncrasies that plague immunoassays when you try to do multiple captures at the protein level. So it's simplicity and throughput and the certainty of the specificity of the assay is what really drove us to capturing peptides instead of proteins.
Now that's what I call a thorough answer. I'm sure you answered his question. Another question, Lee. Is it possible to quantitate peptides from different proteins using the same workflow?
Yeah, that's a very useful question. And the answer is yes. Our objective has been to design a consensus workflow which will allow us to detect and quantitate a wide variety of proteins. Now, the main thing about proteins, as everybody knows, is that they can have widely different physical characteristics. They can have different digestion parameters, etc. So it doesn't go without saying that one should need to test a generic protocol to see how well it's going to work for the particular proteins you're interested in. And if, in fact, you need the maximum possible achievable sensitivity for a particular analyte, there are cases in which it does pay to optimize the procedure a little bit for a particular kind of target. But in general, it's amazingly effective on a very wide variety of targets with this unified consensus protocol that we have implemented on the Bravo robot.
Okay, Lee, and a question for you. What is the ideal length for a peptide against which to develop a SISCAPA assay? How long or how short can you go?
Well, the boundary conditions are what is going to fly well in a mass spectrometer to give you good quantitation, and that's typically in the range of 6 to 22, 24 amino acids. Although we've occasionally used slightly longer ones than that. And the other boundary conditions are you need something which is proteotypic and therefore unique to a particular protein target, and those are usually more than 7 or 8 amino acids long. And finally, you need something that's long enough to generate an antibody against it, which is also on the order of 7 to 8. So the range that we have used practically, I think the shortest we've run is 7 amino acid peptide. And the longest we've used routinely is 22 or 24 amino acids. But that length spectrum comprises a large majority of the tryptic peptides that we might ever want to go after.
Okay, Lee. And a question, what kinetics and other characteristics do you look for in your capture antibodies?
We look for really specific characteristics, as you can imagine, to get the performance that we need to allow the antibody to distinguish one particular peptide out of this mess of hundreds of thousands of other tryptic peptides in a plasma digest, for example. So we look for very high affinity, but at the end of the day, what we use is an approach that screens the antibodies in the actual SISCAPA assays at high throughput, so that we can choose based on performance in the final assay of application. And the primary characteristic that we're selecting for there is most often a slow off rate. And so if the dissociation rate of the peptide from the antibody is long enough to allow the antibody to hang on to that peptide during the extensive washing steps that we implement on the Bravo to remove all the non-specifically bound material, then that's very likely to be a working antibody.
Oh, another great answer, Lee. But unfortunately, we've run out of time for our webinar. But please note that the webinar is going to be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. I want to say thanks again to the panel for their outstanding presentations, and I want to say thank you to our audience for your attention and for your very thoughtful questions about various topics brought up during the webinar. And thank you to Agilent, whose support made this webinar possible. Bye for now.